Hello everyone. Today I'm going to present the practical 2A experiment, which is spectrophotometry sugar analysis using the 3,5-dinitrosalicylic acid DNS method. So this is our group member, which is we have Solomon Diaz and me, Nicholas Chan. So for the objective of this experiment, we should introduce the student the another example to use of spectrophotometry for preparing the glucose standard curve. So the material we need in this experiment is spectrophotometer, hot plate, glass beakers, cuvette, test tube and test tube rack, micropipette and tips, distilled water, standard glucose solution, 10 gram per liter, sodium potassium tartrate, rochette salt, and dinitrosalicylic acid, DNS region. So for the method in part 1, we have number 1, pipette in duplicate the following reagent into series of dry clean and labeled test tube as indicated in the following table. Number 2, after replacing the above mentioned solution in the labeled tube, shake well and then place them in a boiling water bath for 5 to 7 minutes. Next, cool the tubes, add 1 ml of Rochelle salt, then transfer the solution into cuvette. Read the extension, optical density of the colored solution at 575 nanometer using test tube 1 as blank control. Number 4, record the reading and plot the relationship between the optical density and the concentration of glucose solution. See whether there is a linear relationship between the concentration of glucose solution and their corresponding optical densities. Keep the standard curve to determine the glucose concentration in the selector sample, which we use in practical 4. So for the result, this is our result. And this is the graph relationship between the glucose concentration and absorbance at 575 nanometer for practical 2A. So next, practical 2B, determination of sugar in sample using 3,5-dinitrosalicylic acid DNS method. The objective of this experiment is to introduce the student to the use of DNS method for quantifying sugar's concentration in given sample. So the material required in this experiment is DNS region, Rochelle salt, cuvette, test tube and test tube rack, hot plate, glass beakers, distilled water, and micropipette and tips selected drinks, the sample itself. So the method in this experiment is we need to prepare 6 clean and dry test tube. We need to add 1 ml DNS region into all test tube containing the samples as measured according to the above table. And we need to boil the test tube for at least 5 to 7 minutes. After boiling, let the test tube cool down at room temperature. Add 1 ml of Rochelle salt solution into the mixture. So this is the result for this experiment. And this is a graph 2, which is the relationship between glucose concentration at absorbance at 575 nanometer for practical 2B. The conclusion of this experiment is the DNS method successfully demonstrated the use of spectrophotometry to estimate reducing sugar concentration in various samples. By reacting with reducing sugars, 3,5-dinitrosalicylic acid was reduced to 3-amino-5-nitrosalicylic acid, producing a measurable color change. A glucose standard curve was generated from known glucose concentration in practical 2A, and this curve was then used to determine the sugar concentration in unknown drink samples in practical 2B based on their absorbance at 575 nanometer. The experiment confirmed a linear relationship between glucose concentration and also the optical density, which validating the DNS method as an effective quantitative technique for sugar analysis. So for the reflection we get in this experiment is this experiment provided hands-on experience in using spectrophotometry and colorimetric analysis for biochemical quantification. I learned the importance of precise pipetting, consistent sample preparation, and the role of temperature in effective absorbance reading. One challenge was ensuring all samples were cooled properly before measurement to avoid fluctuation in OD values. I also understood on how curves are fundamental in converting raw data into meaningful concentration. Overall, this practical reinforced the link between a chemical reaction and analytical techniques in biochemistry, which is very essential for the application like food quality testing and clinical diagnostic.
And that's
So
it from
that's all
us.
from us.
Thank you
Thank
very
you so
much.
much.